What's up, guys? So today I'll be going over topic 5, which is features of an experiment. First of all, an experiment is when a researcher tries to manipulate certain factors in order to test or isolate the cause and effect of a specific hypothesis or just to describe a fact. So the way that I'm going to be explaining things is by using two examples in order to define and implement all 8 key terms that are in this topic. So the first 5 key terms that I'm going to go over are independent variables, dependent variables, confounding variables, control group, and the experimental group. So for our first example, I will be using plants. This is Lua, this is Chives. We will be experimenting our hypothesis that if we substitute coffee instead of water, then as a result, our plants will grow at a much faster rate. Now for this experiment, we will be having two groups, our experimental group and our control group. Our experimental group is the one that will be receiving the treatment. This is the one that we are going to be tested on, and it's the one that involves the independent variable. On the other hand, our control group will not be receiving any tests and it will not be receiving any treatment. It will just stay the same. Now, our independent variable is the factor that's been manipulated and controlled, which in this case would be the coffee. Since we decided to substitute the coffee for the water, we created that change. We decided to change the liquid that was going to be inside the medium, or the plant. So in this case, the experimental group is the one that is being controlled by us. We decided to pour the coffee instead of the water. Now, the results of the independent variable will determine the dependent variable. So the dependent variable is the outcome due to the manipulation of the independent variable. So in this case, our dependent variable would be the rate of growth of the plant. Depending on the results that we got through whether or not the experimental group grew 2 times faster or compared to the control group, that is our dependent group. Now, there are outside factors that could also potentially affect and impact the results of the experiment. So these outside factors are also known as confounding variables. An example of confounding variables in this case would be the temperature of the atmosphere, or the type of soil that's being used, or the type of plant that is also being experimented on, which in this case pretend that they're both the same plant. But those outside factors could also potentially hinder and skew the results of the experiments. Also remember that the control group and the experimental group serve as a comparison when evaluating the effects of the experiment. So in order for me to define and explain the last 3 key terms of this topic, which are double-blind procedure, random assessment, and placebo effect, I wasn't necessarily able to use the plant experiments since plants can perceive things psychologically, so that wouldn't have worked. So I use the second example now. Let's say that I am a researcher who has developed a pill that could potentially make someone stronger. Now, I want to test this pill out on participants to see whether or not the pill is effective. What I'll do is I will form 2 groups, a control group and an experimental group, and I will keep things constant. So I will use random assignment, and random assignment is basically when participants are randomly put in either the control group or the experimental group, and this is so that I can minimize and prevent any confounding variables or selectional bias. For example, if the participant's ethnicity or their personal preference or such things like that could be examples of confounding variables in this case. So after that, placing them randomly, the experimental group will be the ones who will receive the treatment. They'll be the ones who will receive the actual pill. On the other hand, the control group will receive a placebo. And what a placebo is, an innate substance or a fake substance that is not meant to have an effect on the control group in any way. And the purpose of this is to see whether the results are psychological or based on pure belief. Now, to further prevent any bias from occurring, the researcher, in this case myself, would also be unaware of who is receiving the treatment. This is called double-blind procedure. This is where both the researcher and the participant are unaware of who is receiving the placebo and who is receiving the I hope that this helped you guys out in any type of way. If I messed up some way or missed something out, please let me know. Well,